Hello, guys. So today I'm going to go over notes with you about passive transport, water potential, active transport, all of that good stuff, just so you have it in video form. So starting off with passive transport, we really need to think about that word passive. Passive is not using energy. If you are being passive, okay, you are not using any energy at all. This is where it becomes really, really important to really think about the words that are being used and not overanalyze them. Okay? Do not overanalyze this. Passive transport is not using energy, therefore, we need no ATP. We remember from regular bio, ATP is our energy molecule in our cells, and it's going to start right now becoming very important. Your food through digestion gets turned into ATP. That's what your body needs to actually function. So for passive transport, we don't need any ATP. This is things like diffusion, where we diffuse down what we call a concentration gradient. You guys already know this from regular bio. We did this with a couple different labs. I mean, last year we did it like with our egg lab when food coloring went in our egg and all that good stuff. So it always moves from a high concentration of things to a low concentration of things. For example, if I would have a beaker of water, put a little bit of food coloring in it, that food coloring will eventually diffuse so it's in equilibrium. So there's an equal amount of food coloring all over. Or this morning when I had my coffee, I put some creamer in it, and then the creamer diffused so it's all the same color eventually. So once again, diffusion, if we have two solutes, if we look at the very first image here, we have a bunch of these gold spheres and they are going to go over to the other side. We have a bunch of purple spheres and they're going to go to the other side, but they're not going to make an equal flip. They're going to diffuse until they're in equilibrium and they never stop moving. So if one goes over, then another one goes back. If one goes over, another one goes back. And it's all within equilibrium. And this leads us to our first part of osmosis. Okay, osmosis is the diffusion of water. Osmosis is the diffusion of water. Other things can diffuse, but when we're talking about water, we use the term osmosis. Up here is a classic experiment that's used quite a bit in science. We have a U-tube. In one side, we have a very high concentration of sugar water. On the other side, we have a lower concentration of sugar water. In the middle, we have a selectively permeable membrane. And as you can see in this picture, that sugar molecule is the big green thing. It can't fit through that little tiny hole. That's what selective permeability is. Only some stuff can fit through. However, our water is small enough that our water can squeeze through. So what's going to happen is our water is going to move over to the side that has that really, really high concentration of sugar water. It's going to move in order to dilute that side so it's an equal concentration to the water on the other side. Okay, so the water is going to move to dilute that high concentration of sugar to make sure that they're equal on both sides. This leads to several what we call osmotic solutions. A hypotonic solution, an isotonic solution, and a hypertonic solution. If we start with the isotonic solution right in the middle, this is normal. Iso means to stay the same, like to isolate, like we're staying the same. Isotonic is where the movement of water out and into our cells is equal because the osmolarity of those solutions on either side of that membrane is going to be the same, or it's controlled. If we would put our cells in a hypertonic solution, i.e. very, very salty water, water is going to move out of the cell in order to dilute what's on the outside to try to make it the same concentration of what's on the inside. So our cells are going to shrivel, or if it's a plant cell, it's going to plasmolyze. Because the plant cells can't really completely shrivel because they have that cell wall around them. So they're going to shrink a little bit. And this is what makes like celery kind of wilty if you leave it in the refrigerator for a week or so. Because what happens is the dry condition of that refrigerator, like that cold, dry air, sucks the water out because the water wants it to be the same on either side. This is why at grocery stores they have the mister that they spray on the veggies to keep them nice and crisp. and it actually puts them in what we call a hypotonic solution. So if we look on the other side, water is going to constantly go into that cell because there's more pure water on the outside than the inside. So water is going to go in to try to dilute all of the concentrates that are on that inside of our cell. So understanding water potential is super, super important, and it can be really, really confusing because math looks scary. And I know the first time I saw this too, I was like, what the hot heck are those? What are the cactuses doing in my math? What this is, it looks like a little trident, if you notice. That's actually the Greek symbol for Poseidon, who's the god of the sea. So that's the symbol that we use for water potential. This is where wording, like I said before, gets a little confusing, and we just need to be careful. Water moves from high potential to low potential. If you just replace this word potential with concentration, then we're good, okay? But it's potential because scientists just want to confuse every single one of you. If you ever get confused with these problems, just think water concentration, okay? Water concentration. If I have pure water versus salt water, which one has a higher water concentration? Pure water, okay? Always think about that. Pure water has higher potential than salt water. So when we look at this, we have water potential, which is just Poseidon or psi, and then we have solute potential, which is the osmotic potential of that thing, and the pressure potential. This is more important for plants. We don't really talk about this with animal cells too much because how much pressure that cell is actually putting on the outside of that cell wall. And we also call this turgor pressure or turgor. So pure water would have 0 pressure potential. In plant cells, they can have up to 1 megapascal of pressure because they need to remain rigid. So when we're going through and calculating these things, we use this formula to calculate our solute potential, or our I crit, sometimes people call it. Whereas I is our ionization constant. So this is the number of particles that are made in pure water. All that means is that, I'm looking for stuff that I have around me. In water, for example, if I have glucose, I have C6H12O6. If I put glucose into water, it's not going to break up into carbon and hydrogen and oxygen. It stays as one thing. Therefore, my ionization constant for glucose would be 1 because it stays as one thing if we put it in water. However, if I have something like salt, NaCl, when that goes into water, it turns into a sodium ion and a chlorine ion. So if I put one thing in water and it turns into 2, then my ionization constant is 2, okay? So that's always going to be given to you usually. I will always give it to you. So the next one is C, and that's our molar concentration. Once again, that's always going to be given to you, or you have to solve to find the molar concentration. So that's, for those of you that haven't had chemistry yet, that's just how many sugar molecules per water are in this solution, and it gives you a decimal. R is a constant. We love constants because it's a plug and chug. So that is going to be 0.0831 liter bars per mole K. Okay. T is our temperature, and it's always going to be in Kelvin. So whatever your degrees Celsius are, you have to add that to 273. So for another example, if we are saying that something is at room temperature is usually like 25 degrees Celsius. I would have to add 273. I have to add 273 to that. So room temperature is 298 degrees Kelvin. The reason why we use Kelvin is because we're actually measuring the movement of water, and at 0 Kelvin, that's when all matter ceases to move. Like air molecules will stop in midair and fall to the ground if they're at 0 Kelvin. So if we add solute to the water, it lowers the solute potential and decreases the water potential. Okay, so if we add solute to water, it's going to lower the solute potential, so it makes it more negative. So more negative is more salty. I like to think of it that way. If you have a really negative person, we call them salty sometimes. So really negative solute potential is really salty, and therefore it decreases the water potential. Once again, if we take that word potential, oh my gosh, spelling, concentration, something I can't do right now. So if we change that word to concentration, suddenly it makes sense. So where will water move? It's going to typically move from an area of high concentration to low concentration, or high potential to low potential, low solute to high solute. Okay, low solute to high solute. If we have high solute, that means low water. Low water, high solute. Okay, it's opposite. And it's going to move from an area of high pressure to low pressure. That's how super soakers and squirt gun works. You pressurize the thing that's in it, and that water wants to leave and move to an area of low pressure, would be outside of that squirt gun. So over in here, I want you guys to think about these for a minute. So pause a little bit, and then we'll go over each one. Yay, you unpaused. So which chamber has a lower water potential? So remember, lower water concentration, that would be chamber B. Which chamber has a lower solute potential? Remember, low solute means high water. So that would be B as well. In which direction will osmosis occur? The water is going to move from A to B. If one chamber has a psi of negative 2,000 kilopascals and the other one negative 1,000 kilopascals, which chamber has the higher water potential? That would be the one of negative 1,000. Okay, that would be negative 1,000. So once again, this is really, really important, mostly for plants. And this is actually how plants suck up water. So think of a tree as a giant straw. In the atmosphere, we have a really low water potential or a low water concentration compared to what's in the dirt. So in the dirt, if it's like kind of moistish, like not super dry, there's no drought happening or anything like that. And this is why droughts are very, very bad for trees. So the tree is actually going to soak up water from that ground because that water eventually wants to get up to the air. It's like a giant tube. So a tree basically acts as a giant straw with the atmosphere kind of doing the sucking on it. Kind of super crazy weird. So for a sample problem, I want you guys to try these. We will go over them in class, but I want you guys to try those. All right. Facilitated diffusion. This is different than active transport that we're going to talk about next. And the reason why it's different is because it doesn't use ATP, but it still uses transport proteins. So if we look back to just basic passive transport, that just goes through the membrane. But there are molecules that are too big to actually go through the membrane, so they need a transport protein. These don't use energy. Ions and polar molecules, so glucose uses this, and sometimes water does use this as well. And water specifically uses this through something called an aquaporin. Aquaporin. Aqua water, porin hole, water hole. So this is a channel protein that actually allows the passage of water in and out of our cells in bulk quantities. In your kidneys, where you need to have that bulk transport of water as well, you also have aquaporins, but they were first discovered in plants that live in water in something called the mangrove. So there are certain types of plants that actually their roots are physically in water, and they use these aquaporins to pump out all of the extra water that comes inside of their cells. So here we have a special type of transport protein, which is a carrier protein. Glucose acts like this as well. So they need a very, very special protein in order to get glucose in and out of your cells. And this is activated and turned on and off by your insulin receptors. So people that don't have insulin can't turn these on and off. Therefore, your blood sugar gets really, really high because you can't pump your sugar from your blood into your cells so your body can use it. All right, so I will make active transport into a separate note set. I hope this helped a little bit.